I'd now like to introduce our one speaker of the day who has no connection to Canada. It's a graduate student in the lab of Dr. Aaron Gettler at Stanford University, Lindsay Becker, has been working on something that's a very interesting work in the field in terms of translational work from the laboratory going through towards the clinic as we speak. Something that's quite unique in that it's potentially genetically targeting a possible therapeutic that could have wider applications for sporadic ALS. So really would like to say a warm welcome to Lindsay Becker, and the floor is yours.
Hi, thanks so much for having me here today. I hope that my screen's showing well. So I'm going to go ahead and get started.
Actually, we can't see your screen, Lindsay, sorry.
RNA binding proteins and how interactions among these proteins may be involved in ALS.
Lindsay, actually, we can't see your screen.
As some of the others have mentioned, 97% of ALS patients have TDP-43 pathology and dying populations of neurons. TDP-43 is an RNA binding protein that plays essential roles in RNA metabolism, including splicing. And it's usually in the nucleus, but in ALS patients, it's cleared from the nucleus, where it forms cytoplasmic aggregates that are phosphorylated and detergent insoluble.
Can you, Lindsay, can you happen to hear me?
My screen.
Yeah, we can't see your screen, sorry. Maybe if it's a Mac, we've been having some problems with people maybe needing to just reopen the, but we're not seeing your screen at all right now.
Oh, I'm sorry. Okay, let me.
Ah, there we go. Perfect. Thank you. Okay, it's all yours.
And I'm going to minimize this. Okay, cool. Yeah, sorry. Let me start over. So, as some of the other speakers have mentioned, 97% of ALS patients have TDP-43 pathology and dying populations of neurons. And TDP-43 is usually in the nucleus, and in ALS patients it can be cleared from the nucleus where it forms cytoplasmic aggregates that are phosphorylated and detergent insoluble. And in some patients we also see nuclear inclusions. We still don't know what role this pathology is playing in cell death, so this clearance from the nucleus might suggest a loss of normal TDP-43 function. And it's also possible that these inclusions themselves are toxic, perhaps sequestering TDP-43 and other interacting proteins and RNAs from performing their usual functions. It's also possible that this pathology is just secondary to neuron death and it's not actually causing neuron death. But one major argument against this last hypothesis is that mutations in the gene encoding TDP-43 can actually cause ALS. Over 40 disease-associated mutations have been identified so far. Almost all of them are in this prion-like glycine-rich domain, and some of them have been shown to increase the aggregation propensity of TDP-43, which suggests that inclusion formation may be contributing to toxicity. So this human genetic evidence demonstrates that TDP-43 dysregulation can contribute to neuron death in ALS. And previous researchers in my lab took a pretty unconventional approach to studying neurodegeneration by expressing TDP-43 in yeast. And in yeast, TDP-43 forms these cytoplasmic inclusions and causes dose-dependent toxicity. So here I'm showing you yeast colonies that are all expressing toxic levels of TDP-43. And the previous researchers in my lab were able to use this toxicity phenotype to perform genome-wide screens to find genes that affect TDP-43 toxicity. And one of the most interesting hits was PBP1, which is the yeast homologue of Ataxin 2. So when you knock out PBP1, the yeast are more resilient to TDP-43 toxicity, but if you overexpress PBP1, then the yeast are more sensitive to TDP-43. And like TDP-43, Ataxin 2 is also an RNA binding protein. It interacts extensively with poly-A binding protein, and it has a poly-Q region that's usually about 22 glutamines long. And this was a particularly interesting hit in the yeast screens because expansions to more than 34 glutamines can cause a different neurodegenerative disease called spinocerebellar ataxia type 2. This is where Purkinje neurons in the cerebellum die. And previous researchers in my lab found that intermediate length expansions that aren't quite to this 34 glutamine threshold can increase the risk of ALS. And this has been confirmed in many different studies in ALS populations around the world. So clearly, there is some interaction between TDP-43 and Ataxin 2, and this interaction seems to be involved in ALS. So can we take advantage of this as an ALS therapy? And to test whether this strategy could work in mammalian neurons in vivo, I crossed Ataxin 2 knockout mice with TDP-43 transgenic mice, and those mice overexpress wild-type human TDP-43, specifically in neurons. So as described previously, the TDP-43 transgenic mice had very rapidly progressing disease, here shown in red, and the Ataxin 2 heterozygous mice had significantly increased lifespan, and the Ataxin 2 knockout mice had dramatically improved lifespan, an 80% increase in lifespan over the Ataxin 2 wild-type mice. And the mice aren't just living longer, they're also less impaired. Motor impairment in the Ataxin 2 wild-type mice progressed very quickly, and in the heterozygous and knockout animals, they had a lot less impairment at each age, and we saw similar trends when looking at tremor or kyphosis, which is a bent back that can be caused by neurodegeneration. And we also saw the rate of disease progression was slowed with Ataxin 2 reduction. So this genetic approach worked very well, but we also wanted to test Ataxin 2 reduction in a more therapeutically relevant manner. So to do this, we used antisense oligonucleotides. ASOs are synthetic nucleic acids that have a similar structure to DNA, and they can hybridize specifically with an mRNA target through Watson-Crick base pairing. And the targeted mRNA but not the ASO, is then cleaved by RNase H. And Ionis Pharmaceuticals developed an ASO targeting Ataxin 2 for us, and Brenda Huang in my lab injected this at P1 into the lateral ventricles, just one-time injection, and we saw that even 3 weeks later, we saw a huge decrease in Ataxin 2 levels in the mouse brains, 77%. But we didn't see any change to the human TDP-43 transgene levels, as you would expect. We found that the Ataxin 2 ASO significantly increased lifespan. We saw a 44% increase in lifespan, and we also saw motor improvement in the mice injected with Ataxin 2 ASO. So lowering Ataxin 2 works extremely well in this mouse model. And how do we think Ataxin 2 is modulating TDP-43 toxicity? So TDP-43 and Ataxin 2 both localize to stress granules. These are highly dynamic accumulations of mRNA, RNA binding proteins, and translation machinery. They form whenever translation is stalled in response to various cellular stresses. And when the stress is resolved, the stress granules disassemble and translation resumes. Stress granules are membraneless organelles that behave like liquid droplets within the cell, and phase separation of stress granules from the rest of the cytoplasm concentrates stress granule proteins. And recent studies from multiple groups have shown that concentrating RNA binding proteins and liquid droplets can seed the formation of insoluble fibrils similar to what we see in disease. Many other ALS-associated proteins also go to stress granules or regulate the formation of stress granules. So because of this evidence, it's been hypothesized that stress granules nucleate the pathological TDP-43 aggregates seen in ALS. And in collaboration with Paul Taylor's lab, we found that lowering Ataxin 2 slows the maturation of stress granules. So in cell culture, stress granules, they start out pretty small, and then they gradually fuse to form these larger structures as they mature. And with Ataxin 2 siRNA, we saw smaller, more numerous stress granules at every time point. That's quantified down here. And stress granules the composition changes as they mature. So next we wanted to see if recruitment of TDP-43 was changing since these stress granules are failing to mature. And we found that with Ataxin-2 siRNA, there was a significant decrease in the percentage of stress granules that had TDP-43 positive signal. So this suggests that lowering Ataxin-2 decreases TDP-43 recruitment to stress granules. And so because of that, we wanted to look at the TDP-43 pathology in the mice as well. And this is showing two different types of inclusions that I'm seeing in the TDP-43 transgenic mice. I tested 7 TDP-43 antibodies in these mice, and all of them recognized inclusions in the TDP-43 transgenic animals, but not in their wild-type littermates. And surprisingly, these antibodies differ in the types of inclusions that they recognize. So as I mentioned in the beginning, TDP-43 inclusions in ALS patients tend to be phosphorylated. It's a marker of pathology. And if I look at antibodies that are not phosphorylation specific, or this one phospho-specific antibody 1, then I see nuclear inclusions tend to be pretty big and round. Occasionally they look cytoplasmic, but mostly nuclear. And then with 2 other phospho-specific antibodies, I see cytoplasmic inclusions that are much more numerous and tend to be a little bit smaller on average. Occasionally these 2 different types of inclusions can overlap in the nucleus. The overlap never seems to be complete. So I quantified these 2 different types of inclusions using the phospho-specific antibody 1 and antibody 2. So this is showing the first phospho antibody, and these are spinal cord hemispheres with the dorsal horn to the left. The phospho-specific antibody has a low level of diffuse nuclear staining, and this is darker in the dorsal horn just because there's a really high concentration of these small neuronal nuclei. But if you look at the TDP-43 transgenic animals, you see much stronger phosphorylated TDP-43 staining. And if you go down to the cellular level, you see these big nuclear inclusions that I mentioned in the previous slide, as well as strong diffuse nuclear staining with this antibody. And the Ataxin-2 heterozygous animals had smaller inclusions that were less numerous, and the Ataxin-2 knockouts had very rare inclusions and generally less this phosphoantibody staining. With the next antibody, again, I'm seeing a weak diffuse nuclear signal in wild-type mice, but in the TDP-43 transgenic animals, I see hundreds of inclusions per spinal cord hemisphere. And the Ataxin-2 heterozygous and knockout mice had smaller inclusions that tended to be less numerous. So Ataxin-2 reduction seems to be ameliorating toxicity by decreasing the aggregation propensity of TDP-43. And we think this happens because Ataxin-2 reduction is inhibiting stress granule maturation, and this impairs the nucleation of TDP-43 inclusions. But it's also possible that Ataxin-2 affects TDP-43 inclusions in a more direct way that's independent from stress granules. And just to recap, the reduction of Ataxin-2 in TDP-43 transgenic mice extends lifespan, improves motor performance, and reduces TDP-43 inclusion pathology. And ASOs targeting Ataxin-2 also improves motor function and lifespan in the transgenic mice. And the reduction of Ataxin-2 also decreases the rate of stress granule maturation and TDP-43 recruitment to stress granules. So this work provides further support for investigating Ataxin-2 reduction as an ALS therapy. And more generally, I think it suggests that manipulating stress granules or the biophysical properties of TDP-43 inclusions has promising therapeutic potential in ALS. And quickly, I want to thank my advisor, Aaron Gittler, and Brenda Huang, Gregor Vieri, and Rosanna Ma in my lab, who did a lot with me, and Paul Taylor, and in his lab, James Messing and Hongju Kim.
Thank you, Lindsay, for that. We really appreciate you speaking with us. I do have actually a question for you. It's interesting that you see in the Ataxin-2 knockouts that you see when they're crossed, that the TDP-43 mice, a percentage of them live for a very long time beyond the controls, and then it seems like a percentage of them live a little bit longer, but maybe 50% of it is reduced, or not reduced, but they die, with a little bit of benefit, or at least extension of lifespan. Is there a reason that you can surmise why there would be such a difference between some of them living way longer and some of them that live maybe just a little bit longer?
So you've taken away my screen, but I have another slide here. So what I didn't say before is that we're actually using 2 different independently made lines of Ataxin 2 knockout mice on different genetic backgrounds, and we did find that there was 2 different groups of responders of the Ataxin 2 knockout mice. Some of the mice seemed to be more weak responders and some strong responders, and the genetic background of the mice significantly affected whether the mice were strong responders versus weak responders. But if you look at the mice in isolation, it might look like a small effect just because for the weak responders, because the strong responders are living so long, but even the weak responders, it's still like a 50% increase in lifespan, which is huge.
Sure.
Not a lot of other people have shown that big of an increase. But yeah, the genetic background of mice does play a role, but even if we look at these 2 different lines of Ataxin 2 knockout mice and separately from each other, then we still see significant differences, even in the weak responding line. It's still a big effect. But yeah, it does seem like there is a tail of some mice that lived a lot longer than others, and we don't really know why.
Sure.
But yeah, my theory, that's just speculation, is that it seems like we see in ALS patients that once they start developing symptoms, they have a pretty fast disease progression, especially compared to other neurodegenerative diseases. And this is kind of indicative to me that there must be some threshold effect where cells are able to deal with TDP-43 dysregulation up to a certain point, but once you reach this threshold, then the cells are no longer able to deal with this, and you get a very rapid cell death. So perhaps those the outliers that are living a very long time, they're just never quite reaching this threshold where the majority of cells can't deal with cell death. But they are still impaired, even the ones that are living a very long time, they do eventually develop some impairment.
I see. No, that's great, and I totally agree. Looking at many years of survival curves, that still, even if it had gone straight down from without those really long-living ones, it's still pretty substantial effect, and it's pretty exciting to see what will happen now as this kind of moves forward to early phase trials in the clinic with the ASOs. So yeah, great work, awesome. Thank you so much for speaking, or for joining us for our virtual research forum, and we really appreciate it, and good luck with your work, and thanks. Have a great day.
Yeah, thanks so much for having me.
Okay, great.